Hello, everybody. We're just going to wait a second for everyone to join from the waiting room. It takes just a minute for everyone to add. And then we'll give another minute or so for anyone else to join us who's running a little bit late. All right, we're going to go ahead and get started. Thank you all for joining us today. My name is Matthew Shackley. I'm the secretary of the IMBES trainee board. And yeah, thank you for joining us for this talk with Dr. Harden. We just wanted to remind you a couple things before we begin that following this session, there's another session in 2 weeks on Wednesday, July 7th at 11 o'clock on Zoom with Elena Grigorenko and Michael Skeety. Also, we are hoping again to hold our 7th biennial IMBES conference as originally planned in Montreal, Canada. The dates for that are July 21st to 23rd, 2022. So planning already for next summer. Again, thank you all for joining us today. I'm going to go ahead and introduce our speaker. So our speaker today is Dr. Paige Hardin. Paige Hardin is a full professor in the Department of Psychology at the University of Texas, where she leads the Developmental Behavior Genetics Lab and co-directs the Texas Twin Project. Dr. Harden received her PhD in clinical psychology from the University of Virginia and completed her clinical internship at McLean Hospital, Harvard Medical School before moving to Austin in 2009. She's published over 100 scientific articles on genetic influences on complex human behavior in childhood and adolescence. Her research has been funded by the National Institutes of Health, the Jacobs Foundation, and the Templeton Foundation. Dr. Harden also writes contemporary pieces for outlets such as the New York Times, The Spectator, Vox, and the Boston Globe. In 2017, she was honored with a national award from the American Psychological Association for her distinguished scientific contributions to the study of genetics and human individual differences. Her first book, The Genetic Lottery, Why DNA Matters for Social Equality, will be published by Princeton University Press in September of this year. And I'm sure we're all looking forward to that. So Dr. Harden, take it away.
Thank you, and thank you for having me. I am going to share my screen. Can everyone see that appropriately? Yes, awesome, thank you. Okay, so today I'm going to be talking about some themes from my forthcoming book, The Genetic Lottery, specifically how we can develop what I'm calling an anti-eugenics framework to understand genetic differences and their relationship to social inequalities that we care about. So I'm looking forward to sharing this work with you and getting your comments on it. Okay. And are the slides appropriately advancing? Yes? Can I get a thumbs up from someone? Thank you, Alexa. Okay, so what I'm going to do today is I'm going to do a very fast and furious overview of some high profile research on polygenic scores as they relate to two socially valued human life course outcomes, and that's educational attainment and externalizing behaviors. I'm going to talk about some high profile news claims about what genetics can be used for, and give some thoughts about why people are often alarmed by the direction that this research is going. And then I'm just going to slow it down a little bit and go back into some more methodological details about what are these polygenic scores doing? Where are they coming from? And we can use that as a jumping off point to talk about why I think much of the rhetoric around polygenic scores is misinformed, either in terms of its theory or in terms of its proposed applications. I'm then going to talk about why I think as social scientists, we actually are really well equipped to make sense of polygenic scores, and how we can take lessons from more traditional social science about how to use data for egalitarian policy goals. And I'll give you some preliminary examples of that. Okay, so if you've been following news related to genetics and technology anytime in the last 3 to 5 years, you will have seen headlines like this. For instance, MIT Tech Review said that forecasts of genetic fate just got a lot more accurate, DNA-based polygenic scores. More recently, NBC News had a thing about a new way of predicting which kids will succeed at school. Look at their genes. So what are these new technologies that people are writing about, people are debating? And they are polygenic scores that are derived from large-scale studies called genome-wide association studies. And I'm sure many of you are already familiar with this as a methodology, but just as a really quick overview to make sure we're on the same page. A GWAS is a hypothesis-free scan of hundreds of thousands or even millions of genetic variants that are scattered throughout the genome. They could be focused on a number of different types of variants, but most commonly researchers are focused on what are called SNPs, single nucleotide polymorphisms, which are single DNA letter differences between people. So I might have a C at a particular location, whereas you have a T. These studies are very, very large because they are dealing with a massive multiple testing problem. I'm going to describe to you a study that we recently completed that has an effective sample size of 1.5 million people. And they're typically, although this is changing, restricted to people who are homogenous with regards to their recent genetic ancestry, and most commonly people whose recent genetic ancestors all resided on the continent of Europe. Given the way that we construct race in the US, these people are overwhelmingly likely to be identified as white in terms of their race. So you have a group of people who have some phenotype, and that can be body mass, that could be height, that could be macular degeneration, that could be educational attainment. And you're comparing them to people who are different on that trait to try to find where in the genome do these people who differ in their phenotype, their observed characteristics, also differ in their genotype. So for each one of the measured genetic variants, you have a correlation coefficient. You have some estimate of the strength of its association with whatever phenotype you're studying. And those correlations can be carried forward into a new sample of people in what is called a polygenic score. Sometimes you also see this referred to as a polygenic index or polygenic risk score. And basically those correlations that you've estimated in your original GWAS are now used as a type of scoring key for a new group of people's genomes. So each one of my genetic variants, I can have could have inherited 0, 1, or 2 copies of the minor allele, the less common variant, from my parents. Those are then weighted by the estimated correlation that we've estimated in our previous GWAS and added up across the whole genome to get at a single number. So this is one way to think about this, is researchers' best guess based entirely on someone's DNA of the likelihood of an individual expressing some phenotype, which was the object of the original GWAS. So polygenic scores have a long history in animal agriculture and breeding programs. They have been making inroads within classic clinical medicine. Could we use them as additional screening tools, for instance, for coronary artery disease? But they really sort of hit the fan in psychology with the publication of a paper in Nature Genetics in 2018, where the focus of the original GWAS in a sample of about 1.1 million people, it was not a physical disease. It was not some sort of characteristic like height or weight. It was number of years of education, how far in school someone has gone. So they did their original GWAS, and then they calculated a polygenic score for educational attainment in 2 independent samples of white Americans, the Health and Retirement Study and the National Study of National Study of Adolescent to Adult Health, AdHealth, which many of you have worked with before. So those results are shown on the left-hand side of this graph. And what we see are rates of college completion by quintile of polygenic index. The AdHealth cohort is the more recent cohort. These are participants who were in high school in the mid to late 1990s. And what you can see is that people who were the highest on this polygenic score were graduating from college at 4 to 5 times the same rate as people who were lowest on this polygenic score. My group took the same polygenic score, also in the AdHealth sample, and looked at this earlier in the educational process. So what I'm showing you on the right-hand side of this graph is trajectories of math dropout from 9th grade through 12th grade by polygenic score. So the black dots are people who have taken a math class each year. Again, this is in the 1990s when most US states made math compulsory for only 2 years, versus the polygenic score of the red dots, which were people who dropped out of math. So my colleagues and I have occasionally referred to this as the leaky genetic pipeline. At every point in someone's math taking curriculum, you can see that people who drop out of math are stratified by their polygenic score or polygenic index. This work has taken on sort of new resonance recently with a number of policy debates about tracking acceleration in math classes in secondary school. It's not just education that we can think of as psychologically relevant polygenic scores. Within the realm of psychiatry, the Psychiatric Genetics Consortium and other related groups have done a number of studies identifying genes related to conditions such as depression or bipolar. Recently, my colleagues and I focused more on the externalizing side of the spectrum. So what we did is we aggregated GWAS data on 7 phenotypes for which there was at least 50,000 people available. We ended up with ADHD in childhood, age at first sex, number of sexual partners, self-reported risk tolerance, whether or not someone has engaged in problematic alcohol use, whether or not someone's ever been a smoker, and whether or not someone's ever used cannabis. And what we did is we did a GWAS to identify, are there SNPs that are correlated with this spectrum across a lifespan of behaviors that are associated with problems with self-regulation or difficulty conforming to social norms. We then did what the authors of the Educational Attainment GWAS did, which is that we took the results of our GWAS and used it to calculate a polygenic score in 2 independent samples, again, of white Americans. This is the AdHealth sample and the collaborative study, The Genetics of Alcoholism. There's a lot going on in this slide here, but what I want to call your attention to are the results on the right-hand side of the graph, where we see a number of socially valued outcomes that are of relevance for people who are policymakers, educators, psychologists, in relation to this externalizing polygenic score. So what we see as the polygenic score is non-negligibly capturing variation in outcomes such as whether or not someone's ever used illicit drugs, whether or not they've ever used prescription opioids, whether or not they've ever had contact with the criminal justice system, whether or not they've ever been fired from work. This paper is forthcoming in Nature Neuroscience, I hope in the next 6 weeks or so. Okay, so we have this kind of explosion of large-scale GWAS studies of socially relevant life outcomes and a concomitant rise in the ability to calculate polygenic scores in new data sets. What do we do with that information? And this has been a topic of really hot debate. So on the one hand, you have people like Robert Plomin, a psychologist in the UK, who in his recent book, Blueprint, advocated that we should make these available to, for instance, admissions officers at colleges or prospective employers. A password protected link could make available a set of polygenic scores and your future boss could look at them. Not surprisingly, this was a controversial suggestion, and the sociologist Catherine Bliss responded to it quite strongly in an interview that she gave to MIT Tech Review, which she said, everyone will know who you are, what you're about. To me, this is really scary. A world where people are slotted according to their inborn ability. Well, that's Gattaca, that's eugenics. So I think her use of the word eugenics here is important because it's giving us clues about like what are the sources of consternation, of alarm, of concern that accompany the development of polygenic scores for these kind of socially valued outcomes. Eugenics is actually a really slippery word, and people can use it in a lot of different ways to apply to a lot of different concepts. Genocide can be eugenically motivated, but also selecting a sperm donor who's taller versus shorter can be eugenic. It's a really broad category that covers things that we think of as obviously morally abhorrent to things that actually people are pretty comfortable with. So I think in order to have this conversation about whether or not the development of polygenic scores is eugenics, we need to be a little bit more specific about what we mean by the word eugenic. I've been heavily influenced by the work of the political philosopher Elizabeth Anderson, who described eugenics along with other forms of social oppression as an ideology, and in particular, an ideology that asserts the justice or necessity of basing a social order on a hierarchy of human beings ranked according to intrinsic worth. So the idea here is that the kind of kernel of eugenic ideology is that they're superior and inferior persons where superiority is defined by genes, and that it's then these unequal social relations that can justify inequalities in the distributions of freedoms, including reproductive freedoms, of resources, and of welfare. So again, eugenic ideology can give rise to eugenic policies that range quite widely from, for instance, some people consider pre-implantation genetic screening of embryos to be a form of eugenic policy, all the way up to involuntary sterilization, selective immigration laws, detention, genocide, and interfering with people's reproductive autonomy. So when people look at the development of polygenic scores and they say, this seems like Gattaca to me, this seems like eugenics, I think what they're saying here is, how do we use this work? Is there any way to use this work in a way that does not assert the justice or necessity of a social order, that doesn't classify people into inferior or superior persons? Catherine Bliss is not alone, and she wrote a really interesting book about social science genetics as a field, but several other people, educators, historians, lawyers have expressed similar perspectives of this work is sort of inherently dangerous. There's nothing good that can come out of this. So I've characterized these two sort of opposing positions as eugenic, which I just described as this idea that social inequalities are manifestations of some inexorable, ineluctable natural order. And with that comes suggestions to use polygenic scores to more efficiently categorize people into that order. So can we use polygenic scores to tell someone about where someone should fall in a hierarchy and get them to that place faster? In contrast, we see more on the Bliss side, what I'm calling a gene blindness perspective, which is really the idea that social inequalities shouldn't be linked to genetic or other biological variables, that genetic information should really be avoided, less people be characterized. And to illustrate the gene blindness perspective, I'm going to add my obligatory Jeff Goldblum in Jurassic Park, because I think that it really captures this idea of, just because we can construct a polygenic score that predicts how far someone will go in school doesn't mean that we should. A kind of larger goal of most of my work these days is to attempt to shift this conversation away from these two perspectives, or away from just these two perspectives, eugenics versus gene blindness, and introduce a third perspective, which I'm calling the anti-eugenic perspective. Key to an anti-eugenic perspective is the idea that social systems are fundamentally human constructions, and that they currently work to the benefit of people with certain forms of embodied privilege. And these can be forms of embodied privilege that we can readily see, such as colorism, skin tone privilege, but it can also be forms of embodied privilege that come with our genetic inheritance. And so correspondingly, genetic information can be used rather than entrenching or categorizing people into a hierarchy, used to identify and reduce social inequalities according to these axes of privilege, which might be otherwise sometimes difficult to see. That's a very lofty kind of abstract idea. So I want to talk for the rest of today's talk about kind of one principle from the anti-eugenics perspective that I describe in my book, which is, what should we do with polygenic scores under this framework? In order to understand what we should and shouldn't do, I think we have to go back a step and remember that polygenic scores are based on GWAS, and GWASs are correlations, right? We are essentially fitting a regression line of someone's phenotype through the number of minor alleles that they have inherited from their parents. What that gets is a whole lot of messy processes. So if we observe a correlation in a GWAS between a particular SNP and a complicated human outcome, whether it be education or contact with the criminal justice system or use of opioid drugs, that could come about because of a number of different processes. So these processes could originate in the lifespan of the individual, but still be mediated through what we would think of as social or environmental processes. So for instance, I have a particular set of genes that give me a fearless and bold temperament as a child that elicits harsher course of punishment cycles from my parents, which further entrenches patterns of antisocial behavior. That's originating, perhaps one way to think about it, in the temperament of the child, but it's operating through a potentially modifiable environment, which are parental punishment strategies. This process could be playing out not just within one generation, but across multiple generations. So I inherited my genes from my parents. I could have inherited genes that are associated with education, but they're associated with education because they are the same genes that my parents have, which are associated with their education levels, which is associated with, for instance, living in cleaner, more cohesive neighborhoods, or being able to provide me with better quality early childhood education. So again, it's a genetic association that could come to be via these processes of environmental mediation. And then finally, we don't have to stop at one generation. This is extending backwards and into our ancestral past. And this is a huge, technical, theoretical, methodological problem in GWAS, which is to what extent are the genetic differences that we're picking up actually correlated with broad cultural differences that are interrelated with demographic histories of migration, of culture, of geography. There's progress on that problem, but currently most GWASs are done in samples of distantly related individuals. And so those patterns of correlation that we see between genotype and phenotype are really ambiguous with regards to process. My colleague, former postdoc Laurel Raffington, who I believe is on this call, took the lead in what I think is a paper I'm really proud of that's out this year in Annual Review of Developmental Psychology that describes these processes in more detail about what polygenic scores could actually represent. But what I'm pointing out here is that in a GWAS of distantly related individuals, a genetic association can reflect a number of different processes, and those different processes then get distilled into our polygenic score. What that means about the polygenic score is that I think there's good reasons why we should not use it, as Plowman and others have suggested, for individual selection in educational context or in occupational selection. There's three major reasons for this. You can think of these as the 3 E's. The first is that polygenic scores are error prone. They're capturing at best 10 to 15% of the variance in an outcome, which means that there's massive uncertainty around the predictions for an individual student. The second is what I was just describing, which is that polygenic scores are equivocal regarding mechanism. They're capturing features that we might consider, unfair to select on, such as the level of the environmental advantage of one's parents, right? So we would probably, many of us, recoil at the idea that we should select rich students to go to college more than poor students simply because they're rich, because richness predicts complete in college. But in using a polygenic score, we might be selecting on just that. And then finally, and this is a problem for almost all work for polygenic scores right now, which is they're exclusive. So most GWAS efforts are really centering on people of recent European genetic ancestry. Those people, as I said, are most likely to be identified as white in the US, and that only, if we're talking about US public schools, for instance, that only describes about half of US public school children. So what should we do with them? If we're not using them to select people into educational programs, what are we doing with them? How should we think about them? And here's where I think that the disciplines represented in this organization really have actually a head start in thinking about polygenic scores, because we're used to thinking about what to do with social science variables. So what I'm showing you here is a graph showing you the rate of college completion in the US amongst adults over the age of 25. This is, I believe, 2020 data, divided by quartiles of family income. So the lowest 25% and the highest 25% of students in terms of their family income background. It's a very familiar story in which children from more advantaged homes are 4 times more likely to graduate from college than children from the poorest homes in America. And I just want to stop and think about what we do intuitively when we see a graph like this. So I think there's a number of different things that we're doing in our heads all at the same time when we read a graph like this, when we're thinking about variables such as socioeconomic status. So on the one hand, we might look at this and think this relationship is actually being underestimated because we're just looking at family income, but what about family wealth? What about assets? What about cultural capital? What about the other aspects of environmental privilege that are not captured by measuring family income, particularly in a single year? At the same time, we might recognize that correlation might be an overestimate of what we might consider the kind of econometric causal effect of just giving a family more money, right? So if we just gave families money, would we anticipate that they would show the same sort of concomitant increase in rates of college completion? Probably not, because there's other unmeasured variables that are confounding this relationship between income and college. We also can think that this is a really mechanistically messy association, right? So like, what are the reasons why people who are raised in richer homes are more likely to go on to college? We can describe that at very kind of basic neurobiological mechanisms. How does early advantage shape, white matter development and the developing brain? We can also talk about it sociologically in terms of like, well, colleges are oriented towards, a culture that's more affluent, and people feel less belongingness when they come from first-gen families. We don't think of the mechanistic complexity as undermining the importance of the observation. We look at this and we see that the correlation describes a meaningful population trend, but it's not deterministic for any individual. There are many rich children who fail to go through college. There are many poor children who do very well in college. And finally, and I think this is really important, we think of this correlation, despite it not being perfectly causal, despite its mechanistic complexity, despite the role of measurement error, despite it not being deterministic, we think of it as morally and politically relevant, right? We look at the trends in education in America, and we see there's this, starting point contingency. How wealthy is or affluent is the family that you're born into, as shaping children's life chances and education, over decades to come. And we think of that as a morally and politically important observation about American life. So my suggestion is that we should not be thinking of polygenic scores as some sort of tool for tracking students. We should be thinking of them as social science variables. So this is data from a similar year, looking at the rates of college completion amongst white Americans by polygenic score, polygenic index, rather than family income. And we see a very similar magnitude of association, but I think our interpretive frameworks should also be similar. This is a mechanistically messy, not entirely causal, contaminated by measurement error, non-deterministic association that nonetheless I think is morally and politically relevant for understanding patterns of inequality in America today. This is not my idea. This is a very old ideal. John Rawls, the political philosopher post-World War II, writing in his opus, A Theory of Justice, pointed out that, once we're troubled by the influence of social contingencies, such as how affluent a family a child is born into, on the determination of distributive shares. What is our slice of pie in American society? And education is a very big determinant of that. We are bound on reflection to be bothered by the influence of natural chance. And by natural chance, he's saying, what do you happen to inherit from your parents? From a moral standpoint, the two seem equally arbitrary. So if we're thinking of polygenic scores as another starting point, measure of a starting point contingency like SES, I think we can look to what we already do with regards to SES data to think about how we use flawed social science variables to advance egalitarian goals. And key to that is that some of the best work using SES to advance social equality is using data on individual SES, not to diagnose something about the student, but to diagnose something about the context that the student is in and whether or not that context is equity promoting. Many of you are probably familiar with this data set. It's Sean Reardon's Educational Opportunity Project at Stanford University. This is combining data on every public school district in the US. And what I'm showing you here is data that they've published on the gap between low SES students and high SES students across school districts in America, which is a first step in understanding which districts are equity promoting in terms of narrowing this SES gap in student achievement. On the right-hand side of this graph, I've shown you data from Austin, Texas, where I'm usually located. And you can see that Travis County fares very badly on this index. We are not an equity promoting district. Okay, so can we do a similar sort of conceptual usage of polygenic scores? And I think we can. I think the one place where we can start to use polygenic scores is in terms of not, again, tracking students, but identifying equity promoting social context. Except the equity that I'm talking about here is not narrowing the gap between poor students and rich students, but between students who are genetically at risk for reasons that we don't fully understand for poor educational outcomes versus students who are predicted on the basis of their DNA to succeed in school. An example of this is our work on math course taking. This came out, I keep wanting to say last year, but 2 years ago in Science of Learning, where we're looking at how many optional math classes students took after the end of compulsory math education by level of polygenic score, but also by level of school affluence. And so what we see here is that in the blue line, these are the more affluent schools, even students who are at relatively high genetic risk for dropping out of math are buffered from math dropout. We don't know why that is, but I do want to say that it's not redundant which schools are best at narrowing that family income related differentials and math dropout. It's related, but it's not isomorphic. It's a different layer of inequity, and it's identifying a different set of school districts that are best at closing those gaps. These are naturally occurring or sort of like endogenous variation in context, but we can also think about this in terms of exogenous variation that's imposed by interventions or policy reforms. For instance, and I really like this paper, my colleagues Patrick Turley and his colleagues published a paper in PNAS where they're looking at the effects of a UK educational reform, which mandated that students stay in school for an extra year. And what they found is that the effect of this educational reform on adult health, the average treatment effect was really marginal, but it was particularly beneficial for people who are at high polygenic score risk for high BMI. So this is a different way of thinking about interventions. Among interventions that are effective on average, which are mitigating these genetically associated forms of inequity by preferentially benefiting those who are most at risk. So I said that polygenic scores are social science variables. I would be remiss in not mentioning two things that are special about them. They're social science variables that are, one, time travelers, because the DNA sequence is fixed at conception and immutable over the life course, even though the associations between DNA and things can change as people go through development. The actual DNA sequence doesn't change. So it doesn't matter whether or not you're measuring someone at 50 or 4, you're getting the same DNA sequence. And 2, DNA sequences are randomly assigned to children conditional on their parents' genotype. The conditional on the parents' genotype is important, but often in social sciences, we are swimming in a morass of endogenous variation, and having something that's rooted in this random assignment can be really, really useful. Okay. So I think by using polygenic scores, not as a system for classifying individual students, but rather as a way of seeing which social contexts and which interventions are most effective at closing inequities, that is one way that we can bring this sort of anti-eugenics framework into thinking about doing and applying genetics research. This is one of 5 principles of anti-eugenics work that I talk about in my forthcoming book, which will be out in September. But in the meantime, feel free to email me or DM me with any questions you have about this work. And thank you.
Thank you so much. That was fantastic. So no one has typed any questions in the chat, but if you have any questions for Dr. Harden, feel free to put them in the chat now and I can read them out. It's probably easier than everyone unmuting. John Higgins asks, what is the value of using polygenic risk above and beyond SES?
Yeah, that's a great question. So I'm glad that you phrased it in terms of above and beyond, because oftentimes we can get into this kind of implicit nature nurture binary, we should either use environmental information or genetics, but I'm really thinking of this as an additional layer of information above and beyond what we already know about a student. So the things to keep in mind is that those things are correlated, but they're not correlated at one. They're not isomorphic. So it is giving you an additional piece of information that's not captured by what we're already measuring about a child. I think if we, for certain situations, genetic information isn't going to add that much, right? Like if you have like a really comprehensive psychological assessment of a child's like current academic and cognitive functioning, a polygenic score might not add that much predictive information above that. But you're definitely getting a gain in variance above SES. And in fact, if you look at the 2018 Nature Genetics paper, they have a figure, I think it's in their supplement, where they look at not just what is the kind of zero order R squared of adding SES or adding a polygenic score, but if you already know sort of observed parental education, observed maternal income, that sort of thing, what is the PGS adding above that, and you are getting an incremental R squared of predictive ability above those kind of more classic social science variables. That's a good question.
Zahara Siddiqui asks, how do you think the utility of polygenic scores may change if they were more ethnically diverse in the future?
Oh, I think that would just, I think it would make everything better for a couple of reasons. One is that we are missing from our studies most of the world's global genetic diversity, and with it, our power to identify specific genes that are causally related to outcomes we care about. And we see this even for things that we might think of as phenotypically much simpler than education. So a recent study by Sarah Tishkoff and her colleagues showed how much we can better understand the genetics of skin pigmentation if we just sample more African genomes in our studies. So I think one, just like the genetics discovery, like being able to identify which genetic variants are related to phenotypes with good statistical power. We are shooting ourselves in the foot scientifically by focusing only on this kind of very narrow slice of the globe. The second thing is if we're talking about improving people's lives, whether it be health or education, using polygenic scores, if those technologies are only applicable to people who are, the white kind of historically dominant majority in the US, that has the potential to really further exacerbate health disparities. Alicia Martin is someone from the Broad Institute who's talked about that a lot. And so I think if we're trying to think about like, what is an equitable rollout of technology, it really needs to be not exclusive to just kind of one racial or ethnic group.
Tammy Campbell asks, what do you think motivates people like Plomin who want to use genetic information at the individual level? How do you think his beliefs differ from, you've explained your assumptions and philosophy very clearly. How do you think his differ?
That's a good question. I'm not going to engage in sort of like an armchair speculation about other people's philosophies. I do think there's just something about genetics that activates kind of like a different frame. It activates ideas about determinism and essentialism that aren't scientifically accurate, but are just like really deeply embedded in our dialogues about genetics. And so I think, if you have this kind of semantic network in your head where genes mean biology, means determinism, means essentialism, it's very easy to go from that semantic network to something about using this for individual tracking and selection, right? I mean, this is why like Gattaca was 1997, right, as a movie, but it still is this very common cultural touchstone in conversations about genetics, and that's like an individual selection one. So, I think there's beginning to be some great work on this about, just in our kind of basic biology education, like when we introduce genetics in high school, is Mendel and pea plants and that kind of single gene monogenic deterministic framework really the best frame of mind, given, the complexity of the genetic information that they're likely to encounter in their everyday lives.
James Booth asks, is it the case that genetics can manifest in behavior more in homogeneous environments? If so, what implication does this have for your work?
Yes, I mean, that's obviously true, right? Like if we see at their very basic statistical level, heritabilities and R-squareds are fractions, right? And so anything that's adding more variability in the denominator of that fraction, whether that be developmental noise or environmental variation, is going to reduce our our heritability or our percent variance accounted for by a polygenic score. What that means for research is a couple things. One of those is I feel like we have to be really careful, and I've written about this before in some of my outward facing work. Like I have a book review in The Spectator that comments on this, which is that we really need to be thinking about, we're describing patterns of variation that matter within people of a certain culture at a certain place in time, not some sort of like law of nature that's true across human history or like across culture. And I think just in terms of understanding the scope of what we're doing, I think that's important. At the same time, and I make this point in a recent annual review of psychology article that I wrote, there's lots of statistics that are specific to a certain historical moment, a certain culture and time, such as the Gini index. And that's describing something about like wealth inequality now in America. That could change over time. That will be different across cultures, that will be different across political economies, but it's still telling us something valuable about the moment that we're living in. And so I think keeping both of those frames of reference, this is like not true of all people, like across all time, but it is telling us something about the moment that we're living in, is the kind of dual frame of reference I'm trying to keep in mind here.
Sophia Carosa asks, who do you think bears the responsibility for ethical use of GWAS, especially when it comes to promoting equity in life course outcomes like education? And in the spirit of your forthcoming book, how should we as scientists educate the general public?
I mean, I think it's really hard to think about any sort of one set of stakeholders for ethical responsibility. I think this is really shared ethical decision making, and some onus is on educators, on policymakers, on funders, on the scientists themselves. Um, and this is something like I'm part of a working group right now that's specifically devoted to what are the scopes of ethical responsibility for scientists to do this work just in the form of communication. And it's been interesting to me that these are people who are, all care about this topic, right? Like, are all the sort of people that spend time on a bioethics working group about responsible science communication. And even then there's really like deep pockets of disagreement about where the boundaries of people's responsibilities lie. That is a long way of saying that, like, I don't have a clear answer for that question. I think this is, I think we are in that time of like a technology upheaval, which kind of like throws a lot of the values up in the air, and people and different stakeholders are kind of trying to articulate a bunch of different competing values. And it will be really interesting to see how that messy collective decision-making process kind of shakes out in the next 5 years or so.
There's a couple of questions here from Emma Hart and Luciano Seed asking about epigenetic factors and asking how differential life events could affect epigenetic factors and how that plays into your model.
Yeah, so I will say that we also do work in this area, and Laurel, who led that paper on, avoiding biodeterminism and our use of polygenic scores, she also led a paper from our group that's just out in pediatrics looking at patterns of DNA methylation in children and how DNA methylation patterns associated with not advanced biological aging, but more rapid biological aging are associated with socioeconomic disadvantage in our sample. So all of that to say, I don't think that gene sequence variation is the end all be all in terms of layers of information you can have about someone's life. I think the most interesting studies are the ones that combine these layers of information. So we have this genetic lottery of genetic inheritance. We have these social exposures. They combine to shape our epigenetics. To what extent can we then use those as tools for seeing the early effects of exposures long before kind of like the long-term adult health sequelae of those early social determinants of health are manifested. So in my mind, what's one of the things that's interesting about both about epigenetic data and genetic data is their use as time travelers, their utility at taking different parts of the lifespan and projecting them onto like a molecular measure that we can then use to look at biological aging in childhood, or risk for high BMI in people who were studied as adults, even though they experienced an educational reform when they were 14. So again, it's adding layers of omics information that really I think is going to produce the most valuable science.
Martha Farah asks, genes presumably affect behavioral outcomes through the brain. She's wondering if there is any additional clarity on causal mechanisms to be had by adding neuroscience data to the mix.
Yeah, so you did something really interesting there, which is that you combine the phrase causal mechanisms. And I think it is the combination of genetic information, which can give us some causal purchase because genotypes are randomly assigned contingent on parental genotypes, and neurobiological information, which can get at mechanism in terms of, okay, well, we know that these genes that you inherit at conception are associated with your risk of using prescription opioid drugs at age 25. What's happening in the lifespan in that intervening time. So if you think about like the ABCD study, right? So how are those initial genetic liabilities related to altered patterns of brain development during adolescence that might make someone more responsive to the rewarding effects of drugs, for instance? And I think that kind of multimodal research is in its infancy, and it's going to be so exciting to see how it unfolds.
Genevieve Allaire-Duquet has a question asking, what could a teacher tell a student that says, I'm never going to be good enough at math because my genetics, why bother? And in other words, how can teachers approach the discussion about genetics with their students?
Yeah. So I will say Lucas Matthews, who's a former postdoc of my PhD advisor, is Eric Turkheimer, has a new, I don't know if it's published yet, preprint on potential for stigmatization amongst students when they've gotten results from their polygenic scores. So there's beginning to be, excuse me, empirically grounded research exactly on this topic. You know, I think this comes back to the frames that are activated in people's minds. Like Richard Dawkins in one of his books was like, what did genetics do to deserve such a sinister juggernaut type recommendation? And, you know, his own work might be at fault for some of that reputation, but this idea of genes as like always collapsing human agency or limiting human agency as being deterministic is a big part of the frame that I'm trying to overcome with a lot of my work. Math is a skill. Math is a skill that responds to practice. And we see that even in looking at the variation in math outcomes that we see for an individual with any given polygenic score, right? So if you look at, you know, a graph of educational attainment by polygenic score, you can run your finger up it and you'll have people who dropped out of high school math and people who have a STEM PhD. So there's always the room for skilled practice in terms of, you know, anything, especially math. It is counteracting these frames of genetics as collapsing our scope for improvement, collapsing our agency, collapsing the value of practice that I think are so perniciously sticky in these conversations.
So to insert a question of my own here, as a teacher education researcher, sort of along those same lines, do you foresee any benefit to bringing up these topics with prospective teachers, pre-service teachers? Like what could teachers do in the classroom with this knowledge?
I mean, I think of, oh, that's a really good. So do you mean like what could teachers do in the classroom in terms of teaching about genetics, or what would teachers do differently in the classroom, given that we have genetic information more generally?
The second part, whether or not they know specific student genetic information, but just with a knowledge of heritability and genetic differences.
Yeah, I mean, I don't really foresee that being the value out of genetics in any near term scenario, which is like, you know, you get a report on your kid, you know, the students in your classroom's genotypes. Like, I just don't foresee that being useful. I feel like where this is useful for education is, you know, just designing effective or more effective teacher practice curricula, educational interventions is a remarkably difficult process. And, you know, as you know, most of the things that we try in education, like, don't make a difference, like don't have an average treatment effect that's like marginally bigger than zero. And so when I think of the value add of genetics for educators, it's not this little Johnny has a lower polygenic score than little Timmy. It's educational researchers waste less time trying to intervene on things because they have been, they are able to sift through correlates of better educational performance to identify potentially effective targets of intervention more quickly, because the basic educational research has controlled for genetic heterogeneity between students. That's where I think that the value add for educators is going to be. Not, I'm going to treat little Timmy different than little Johnny, but on average, this practice is better than this practice. And I know that on the basis of an RCT that was more effective because it chose an intervention target based on more causally rigorous research, right? So it's more at the level of intervention research than differential practice or differentiation in the classroom.
Thank you. Sort of shifting then, Lucia, I apologize to people in the chat if I'm mispronouncing your names. I'm trying my best here. Luciana Troiani asks about policy. What kind of policies do you think could be implemented based on Anti-Eugenics view?
Yeah, so this kind of goes back to, you know, I think there's a really broad scope for this. I think one thing that I want to emphasize here, because I think it's missing, or easily overlooked in these conversations is that developing effective policies and interventions is really hard to do. Like looking at the vast matrix of things that are correlated with other things and identifying this is the spot in this web that I'm going to push on to make a difference, and push on to make a difference for people who are, you know, not most likely to be helped by anything, you know, especially if we're talking about avoiding Matthew effects. It's a really difficult scientific problem. So I really do believe that the biggest value add for genetics in terms of policy is not going to be policies about genetics, but simply integrating genetic information into the search for environmental causes, to make those studies more rigorous and less confounded by heterogeneity between people. That's like, if no one else did anything but just do the research you're already doing and control more rigorously for genetic differences between people, like I would consider this work to be a success.
Aliva asks more of a general question. Could you please explain heritability for those of us who don't study genetics? Tend to find this term elusive and very confusing.
Yeah, so heritability is an estimate of how much the observed phenotypic differences between people that you see are due to genetic differences between people. It's necessarily a ratio. So if you have very varied environments, then the pool of variation in phenotype is going to be bigger and heritability is going to be smaller. And heritabilities can be, you know, it's about genetic differences are manifest as phenotypic differences, but these can operate through social processes. So the classic one for Anti-Eugenics is that, you know, if we didn't let redheaded children go to school, then genetic differences between people in hair color would come to be correlated with differences in their literacy, and that would be manifested in the heritability. So it's at its core about to what extent are the observed phenotypic differences that you see between people due to genetic differences in their inherited DNA sequence. That's what heritability is.
Tammy Campbell has another question. Do you think there isn't any argument for individuals having access to their predictive information to make choices? For example, if I have a higher chance of being good at maths than music, could I choose to pursue maths, or might this become a slippery slope?
Yeah, I mean, I have to say that, like, my prior is that, like, whatever information people want about themselves, they should have access to. I mean, like, people make decisions on their horoscope, right? Like, is, you know, and that, I don't think that's a good decision necessarily to be like, I'm not going to do something today because I'm a Taurus and it's Mercury retrograde, but we don't say you're not allowed to do it. I think it comes down to an appropriate amount of science, education, and communication about what the limits are of taking that information as really deterministic about your outcomes. That being said, like, this is why 23andMe exists, right? Like, people love, like, why do people want reports that say you have a 95% chance of green eyes? Like, they can look in the mirror and see that they have green eyes, but they still want that information about themselves. And I think that kind of, like, desire to know yourself can't be underestimated.
Thank you. There's no more questions at this time, so we'll give it another 30 seconds or so if anyone wants to chime in with anything else for Dr. Harden.
Can I add one more thing? Which is that a couple people have asked on Twitter and elsewhere about potentially using the book for class. If you're interested, if you want to see a table of contents and you're thinking about using for a class, just email me and I can send you a table of contents. And also, it's really important to me personally to always make sure that my students have access to free or low cost materials. So if you're thinking about assigning this for your class and want to have a solution to provide a free copy rather than asking your students to buy it, that's also potentially something that we can work out. So please just email me about that.
Fantastic. Well, thank you, Dr. Harden.
Thank you all. Those were great questions. I really enjoyed it.
We're going to shift gears now, and we're going to introduce Sophia Carrozza. As part of the virtual conference series this summer, the IMBES trainee board hosted or called for submissions of what we called elevator pitches, or a pitch competition of short 5-minute videos of trainee scholars in the field of MBE. Sophia Carrozza won our pitch competition as judged by the IMBES trainee board, and she's here with us today to introduce herself and share with you her 5-minute pitch video. Sophia.
Thanks, Matthew. Thank you so much, Dr. Harden, for a really interesting talk, and I'm looking forward to reading your book. And I'm excited to be able to share with everyone today my research pitch, the project from my first year here at the University of Cambridge, where I'm a PhD student and a Marshall Scholar at the Cognition and Brain Sciences unit. And as you'll hear, I'm interested in the effects of abuse and neglect on the developing mind and brain. So I'll just share my screen now.
In 1995, Vincent Felitti and Robert Anda made a startling discovery that would affect a revolution in the fields of public health and developmental science. That children who experience adversity in the home are much more likely to later suffer from a wide range of illnesses, both physical and mental, as well as outcomes like poor educational attainment, addiction, and incarceration. This discovery rightly provoked a great deal of interest in research, because it revealed that if we stop the cycle of adversity, we could build a more just society in which all children can flourish. But in the nearly 3 decades since this original study, the field still hasn't reached consensus on the nature of early adverse experiences, let alone the mechanism through which they interact with a child's developing mind and brain. In part, this confusion is due to methodological issues, like small sample sizes and inadequate controls. But the issues are also theoretical. Is it the cumulative number of adversities that matters, their frequency or duration, or is it the age at which they occur? These questions aren't just idle disagreements between academics. They have real-life implications for trauma-informed care, whether that's reforming the juvenile justice system, improving mental health treatments, or implementing interventions for children in the classroom. So it's essential that we get this right. The goal of my research is to formulate a clear and coherent account of early life adversity. In my most recent project, I set out to find how different forms of adversity relate to one another and to cognition and emotional functioning in adolescence. My aim was to test an influential theory that posits deprivation and threat as 2 separate dimensions of early life adversity. I first obtained data from ALSPAC, which is a large cohort study here in the UK, not only because of the size of this sample, but because it has prospective measures of adversity collected across childhood. They're measured 5 times between birth and age 7, and include things like physical and sexual abuse, financial difficulties, and neglect. As for outcomes, I looked at externalizing and internalizing symptoms, as well as measures of cognition in adolescence. Then I fit a mixed graphical model to the variables, a type of network that allowed me to account for the complex set of interactions that exists between these data. Then I identified groups of densely interconnected nodes, also known as clusters, to see if there were preferential links between certain kinds of early adversity and certain outcomes. Finally, I tested whether the timing of the adverse experiences mattered, using a technique called model-based recursive partitioning, which identifies optimal splits in the data based on a moderating variable, in this case, the child's age when the adversity occurred. So what did I find? Well, in a network of adversity variables alone, we get these 2 distinct clusters of experiences. The one on the bottom is composed of situations of threat, times when the survival and well-being of the child was threatened by things like domestic violence or emotional cruelty. And the one on the top is composed of situations of deprivation, when the child didn't receive what he or she needed to thrive, like times of financial difficulties or neglect. So it seems like there are these two distinct categories of early life experience. And when we introduce adolescent cognition and emotional functioning into the network, all of these outcome variables cluster with the deprivation group and none with the threat group. What about when we consider the timing of these experiences? Well, the algorithm splits the network into three, one for infancy, one for early childhood, and one for middle childhood. And in each stage, adolescent scores of cognition and emotion cluster with measures of early deprivation. The specific form of the deprivation varies, from parental separation and caregiver instability during infancy to a broader range in early childhood, and later on, maternal neglect. So this data-driven analysis revealed that early adversity tends to occur in patterns that line up with deprivation and threats. But rather than specifically affecting cognitive and emotional development, a wider range of downstream outcomes appear to be closely related to experiences of deprivation across childhood. Following up on these findings in my next project, I'm using MRI data to analyze structural features of the children's brains, to test which changes in white matter organization may be a stepping stone from early adversity to these later difficulties. But I think we know enough already to make concrete policy recommendations. If we want to mitigate the effects of early adversity, a really promising place to start is this deprivation cluster. Legislators and leaders should ensure that parents have the socioeconomic security necessary to care for their kids, both materially and in terms of the time and love that they want to show them. Thanks for watching.
Thank you, Sophia. As you can see, well deserving of being the winner of our pitch competition. We have 10, 15 minutes for questions for Sophia or comments if anyone would like to chime in with anything. So we'll just take the same format as before. If you have anything that you would like to put in the chat, we can ask Sophia. Wow.
Rome is in my group, so she's biased.
Okay. I would jump in with a question, but I cannot begin to get my head around the modeling that you use there. Do you have any background in statistical stuff, or I was way out of my depth there.
Oh, yeah. Well, hopefully I can, learn how to translate it a little bit better. But yeah, I do some computational methods here. I did some for my master's, so that gave me a place to start with my PhD. But yeah.
James Booth asks, do you have any ideas of what threat may be related to in terms of outcomes, something that was not measured?
Yeah, definitely. So one of the things that I found interesting about Dr. Harden's talk is some of the things that big data can reveal that maybe we don't have a good understanding of from smaller studies. And this has been a consistent theme in studies of threat, that smaller studies have found associations that don't hold up for a number of reasons, presumably in larger studies. So in the same data set, there was another study that looked to replicate the finding that early threat is related to later difficulties with recognizing emotional faces. So this a bias to interpret faces as angry, and they found no evidence of that. So again, maybe for a different reason, but it could be that with increased power, we're finding that previous relationships are not actually founded in the data. But it does seem that that there are associations with poor mental health. But this is something I am interested in looking at further, what specifically might be linked to early threat.
Thank you, Sophia. We'll take another few seconds here and see if anyone else has anything else they would like to ask or contribute. Luciano Seed asks, has this work been published yet? Is it in the pipeline and they want to read it?
Thank you. That's very kind. Yeah, it's about to be submitted, and I will be definitely posting a preprint and a copy as well if and when it's accepted. But yeah, I'm looking forward to that.
Where will you be posting that? How can we find that?
I'm on the OSF, and yeah, if you search my name, it'll come up.
Perfect.
Thank you.
Rachel Romeo says, apologies if I misunderstood this, but how does your model account for when children experience multiple adversities, say both deprivation and threat?
No, that's a fantastic question. Yeah, it's the reason that I use the network model, because it's really hard to do, but it's so important given how common co-occurring adversities are. So the model uses node-based regression. So essentially, you're using each node in the network to predict the score on one node at a time. And in doing that, you can get a measure of, in that sense, it's almost like a partial correlation in that you're controlling for all of the other variables in predicting from one node to the next what specifically that relationship is, holding everything else constant. And so in that sense, once you put all of the nodes into the same network, then you get a general picture of how these are related to one another. So the disadvantage then is that you can't draw as clear conclusions because there are so many complex interrelations. And that's why I went with the clustering to try to get a sense of what the overarching pattern was among these different forms of abuse and deprivation. But yeah, if you come up with another method that works even better, I will be very open to it because it's a thorny and perplexing issue.
John Higgins asks, can you tell which is easier to bounce back from, deprivation or threat?
Yeah, that's an interesting question. I think the field is focusing a lot on resilience now, which can be interpreted as bouncing back or recovering from what you've experienced. I haven't seen any comparative studies, and I think it's hard to sort of measure the degree of adversity across different forms and sort of assess the severity of that, and which would then allow us to sort of examine how much resilience is needed in every context. But I am very hopeful that these kinds of studies can help us identify what factors make it easier to bounce back. So what sources of social support and other things can help you remain strong and recover from experiences of adversity. Yeah.
All right. Oh, we had someone pop up there, but all right. Well, if no one has any more questions for Sophia, thank you so much, Sophia. Congratulations.
Thank you, everyone.
Her video on the IMBES YouTube as well. So if you would like to rewatch it or rewatch any of this, I believe it's going to be hosted on the IMBES YouTube channel. All right. I'd just like to remind everyone once more that, oh no, I can't make it bigger. Hold on. Once more that in 2 weeks, our next session on a Wednesday, also at 11, with Elena Grigorenko and Michael Skeety. And then also next summer, as we had planned in Montreal, Canada, July 21st to 23rd, our 7th biennial IMBES conference. We hope to see you all there. Thank you everyone for attending. Have a great rest of your day.